Okay, I am going to be so punctual now that I heard the bell. Hi, I'm Kelsie. I'm a PhD student at Harvard University, and I'm excited to talk to you today about structural variation in Hemitrichus margarita saeventer. So a pangenome is a collection of the complete set of genomic elements that occur within a species or clade, and a pangenome graph integrates the assembly of multiple linear reference genomes while preserving the coordinates of a linear reference genome. In this pangenome graph, nodes are these oriented DNA segments, edges show the connectivity relationships between these nodes, and we use bubbles to characterize variants for the different individual haplotypes, which are represented by the dotted path lines. And they're really powerful tools for identifying many different types of structural variants. When we scale this up, pangenome graphs have the power to reveal thousands of structural variants and DNA sequences that would be undetectable with traditional reference-based methods. And this allows us to build a better reference that captures the genetic diversity of a species or clade, and can also enhance our understanding of structural variation. And structural variants have been receiving a lot of recent attention, especially for having important roles in adaptation, and they often directly or indirectly underlie fitness relevant traits. But their average effects on fitness are not as well understood. So I focus on the open vegetation biomes and seasonally dry tropical forests of South America, the largest patch being this dry diagonal region in Brazil that has these dry shrublands of the Caatinga and savannas of the Cerrado. And these are one of the most understudied but threatened habitats on Earth. They're all seasonally stressed by drought, have high levels of endemism, but have received much less research attention than tropical humid forests. And evaluating these questions in this system is really important from both an evolutionary perspective as well as a conservation and climate change perspective, as it can help enable an understanding into the genetic basis of adaptation to arid environments. My focal species is the pearly-vented toady tyrant, Hemitrichus margarita saeventer. The Hemitrichus genus is comprised of 22 species, all found in humid forests, but this is the only member of the genus that is found in these dry forests. And it's also a subassine, which is a suborder of about 1,000 primarily neotropical birds, and they're super common and diverse. About 1 in 3 birds that you'll encounter in the neotropics is a subassine. But despite their tremendous diversity and abundance, they're relatively poorly studied in comparison with their sister clade, the Aussine passerines, and especially underrepresented in genomic studies. So my questions are, how extensive is structural variation in the species? Where are structural variants located in the genome? And what are the fitness consequences of structural variants? So I sequenced 5 birds collected from the Caatinga and 1 outgroup, vermilion flycatcher, at about 30x coverage per bird using PacBio hi-fi circular consensus sequencing, which gives really highly accurate long reads. And then I used hi-fi ASM, which is a haplotype resolved de novo assembler. So even though I sequenced 5 birds, I really have 10 haplotypes. And all my assemblies were really good quality. All of them had less than 1,000 contigs, and the average N50 was 17.4 megabases. So we first generated a chromosome-level reference assembly to aid in the pangenome analysis. And I've been working on this with Dr. Clarissa Ferreira de Carvalho, and I would tell you to go to her talk, but it's happening right now in the diversification session, so you have to look for a recording. But we used high-C data to scaffold our assembly from around 600 contigs into 34 chromosomes. Then we used synteny mapping to validate our chromosomes, and so we mapped it to the closest available reference genome, which is Queerschifia lanceolata. And because this was pretty synthetic with the Queerschifia, we were very happy with our chromosomes. But we actually found a potential fusion between chromosomes 3 and 4 of the Queerschifia genome. It's this light green and blue at the top, which seems to have fused into our chromosome 1. And we're looking more into this putative fusion, but it could be really cool. So we have to really validate this more. So I used the pangenome graph builder PGGB, which creates unbiased pangenome graphs. And I first partitioned the sequences into 32 communities or autosomes. And in brief, the main steps are performing pairwise sequence alignments, a graph induction step, and then a progressive normalization of the graphs. Then I decompose the graphs into a VCF format and classify the variants as SNPs. Indels I call less than 50 base pairs, and structural variants are larger than 50 base pairs. And then also use several programs to aid in inversion discovery. Minigraph is another graph builder, and SVIM and Siri are assembly-based structural variant callers. So across the genome, I found about 6.5 million SNPs, about 1.7 million indels, and about 100,000 structural variants. The structural variants cover 3 times more base pairs than SNPs. Indels cover 4 times more base pairs than SNPs. And they tend to be more densely located towards the end of the chromosomes, probably near the telomeres. And looking at each type of deletion insertion, within indels, deletions are the most common type of variant, and within larger structural variants, insertions are more common. And looking at the size distribution of structural variants, this ranges from 50 base pairs to 10 kb. And I first wanted to look and see what's going on within these different structural variants. So I looked at the repeat content, and I found that over 50% of structural variants overlap with repeats. This is compared to repeat content in the hemitrichus being about 14%. So the biggest majority is simple repeats, but then next, like the 11% span multiple types of repeats. And this, like, structural variants are thought to be formed more frequently in repetitive regions due to errors in DNA replication and repair, and this kind of supports that. Then using several structural variant callers, I found 154 unique inversions ranging in size from 50 base pairs to half a megabase pair. And Siri detected much larger inversions. Note the differences in the axes. It was just like off the chart of the other ones when I tried to combine them. And I'm looking now into trying to understand where the inversions are and what might be underneath them. But I didn't want to get like hand wavy at this conference yet. So I'm looking at these right now, and this is really cool. So to understand the impact of genome-wide variants, I first constructed the unfolded site frequency spectrum of the different variants in multiple genomic regions. So coding sequences, CDS, regulatory regions, the untranslated 5' and 3' untranslated region, and introns. And all the variant types are concentrated at low derived allele frequencies, but indels and structural variants have more rare variants than SNPs do. And this is especially more evident in the coding regions. Then looked at the distribution of fitness effects using FastDFE, which is a maximum likelihood program that uses the site frequency spectrum of the variants and compares it to a putatively neutral site frequency spectrum. And the DFE estimates this variant deleteriousness based on the population-scaled selection coefficient, which is a function of the effective population size and the selection coefficient. And I found that indels and structural variants are more deleterious than SNPs in both coding regions and introns, but structural variants are much more strongly deleterious in coding regions. And what we're really excited about is looking at the potential role of structural variants in adaptation to these dry forests. So we currently have whole genome resequencing data from Margarita Saventer across its range. And Dr. Havana Lima is looking at phylogeography in Hemitrichus, Margarita Saventer. And Clarissa is using PopGen data to compare it to its 2 closest relatives, Striaticollis, which has an Amazonian distribution, and Nijipendulus, which has an Atlantic forest distribution. And we also have transcriptome data from 3 males of each of the 3 species to explore potential gene expression differences between the species from physiologically and metabolically important tissues like kidney, liver, and muscle. And we can integrate this with the structural variant data to genotype any structural variants across the species range and between the species. And also, which is really exciting, is we might be able to see how structural variants influence gene expression, especially given the overall fitness effects I found with the different variant types. So in conclusion, pangenome graphs are really powerful tools to reveal all different types of structural variation, especially considering I only had 5 birds or 10 haplotypes. Most of the structural variants overlap with repeats, which likely facilitate structural variant formation, or could be from TE insertions. And structural variants are more strongly deleterious than other variants, especially in coding regions. And that I want to thank my lab. I especially want to thank Bohau and Scott, who have been pioneering the pangenome analyses and taught me a lot. And my amazing collaborators in Brazil. And yeah, thank you. I have time. Yeah.
Great talk. I was just wondering about the complex structural variant category and if that can be easily decomposed further into some nice categories, or it's kind of really just a jumble of everything.
So before I classified them, I already did a step. So I saw your talk. It was really cool too. And there's like nested variants which are inside like other variants. I broke that down first. But those complex types, we can't tell if it's an insertion or deletion based on the genotype or the outgroup, which we use to polarize. Yeah.
So if I'm understanding this right, with just these 5 birds, there are like multiple sections of the genome of at least a few kb that are flipped, within those 10 haplotypes. Is that like a surprisingly high number to you? Do you think that's the case in all birds? And we just haven't been finding it?
Based on like the 2 other bird pangenome studies I've seen, if I scale up my sample size to those, it kind of checks out. But I think like the size of the inversions really depends. It might be more relevant if it's like proportional to the size of the chromosome it's on, because birds have microchromosomes. But I think, yeah, there could be like a ton of this missing in bird genomes, and it could be very prevalent. Hi, Flavia.
Do you have any guess of what type of transposable element could be more like involved in this like structural variant?
There's this peak, which I think are lines, around like 6,000 base pairs of the structural variants, and I have to like look at the extract the variants and look at repeats like on the variant itself, but it might be like lines. Well, that's like the 3rd most common type I found.
So hello, everyone. Good morning. My name is Matt Hufford, and I'm from Iowa State University. And today I'm going to be talking to you about genome evolution post-polyploidy in maize and its wild relatives. And I want to start off by saying I love coming to the evolution meetings because I get to hear about all these amazing systems, and then I come and talk about corn. And this is my not very charismatic system, which is, lined up rows and perfectly even. Doesn't look like there's a lot of variation there. But I promise you that corn is not boring, and there's some really interesting evolutionary history in its background. And I first got turned on to the system when I was a graduate student at the University of California, Davis. And I took a wonderful agricultural ecology course by Louise Jackson, an amazing professor at UC Davis, where she told us about the domestication history of corn. And for the longest time, people didn't know what the wild progenitor of corn was, because the morphological differences between teosinte, which is the wild relative, and corn are so dramatic. So if you look up there in that figure, in terms of the architecture of the plant from teosinte to maize, you see this reduction in branches during the process of domestication. And if you look at the ears, they're really dramatically different. So in teosinte, you have these disarticulating seeds which break apart and can disperse. That makes a lot of sense for a wild plant. But within maize, you have all of these naked kernels that are bound to an ear, which is great for a domesticated crop. So major morphological differences during the process of domestication. And because of these, for a long time, people fought over what the progenitor of maize was. So you can see those two guys down there shaking hands in that figure. So that's Paul Mangelsdorf and George Beadle. And they had this bitter battle of what was the progenitor of maize in the domestication story. And Beadle actually rightly inferred that it was this teosinte that had major morphological differences and that farmers had selected on these changes. And he did mapping populations between teosinte and maize crosses and found that as few as 5 major loci could explain the differences between maize and teosinte. And so John Dobley up there in the right-hand corner spent his career trying to find these 5 major loci that explain these differences between maize and teosinte, and he found several of these. One of the most famous ones is the TB1, teosinte branched 1 gene. And what he discovered was that that explained most of the difference in terms of the plant architecture, and that about 20% of the difference was due to the effects of this transposable element that inserted upstream of the TB1 gene, increased the expression of that gene, act as an enhancer, and then suppress those lateral branches within maize. So he's able to get it down to that single gene level. So I was hooked after hearing about this domestication story, and so I spent my PhD down in Mexico when I was primarily an ecological and conservation geneticist, mapping these populations of teosinte in Mexico. And then trying to understand how human influences on these populations, reduction in population size, affected the levels of genetic diversity and the amount of genetic load within these populations. So I've arrayed some populations in this plot up in the top right, where you can see going from a Hue to SLO. These are decreasing genetic diversity in these populations and increasing genetic load. And then I connected the amount of genetic load within these populations to the fitness of these plants as well. But then as I became a postdoc and a faculty member at Iowa State, I became more interested in genetic diversity in maize, how that's changed over the evolution of the crop, and looked primarily first at this domestication history. So if you think of these farmers in early Mexico that were developing maize, when they were out there selecting from teosinte, they probably weren't selecting from every single plant that's distributed across Mexico to form this crop. There's probably certain populations that had interesting morphological innovations that they primarily selected on, and that results in a genetic bottleneck. So I looked at these different populations of teosinte, compared that to maize, and documented the genetic bottleneck during domestication. And then we studied the process of adaptation of maize. So maize moved from a lowland center of origin, where it was domesticated from this Parviglumis teosinte, and then it moved up elevation to the central plateau of Mexico, and it encountered this high elevation teosinte, a different subspecies of teosinte, and received gene flow from that high elevation teosinte, and specifically inversion polymorphisms that flowed into maize and conferred adaptation to these high elevation regions. So exciting things that we did just with resequencing and SNP data, but then over the last 3 or 4 years, genome assembly has become possible with maize, which has about a 3 gigabase genome. It's about 85% repeats. So difficult genome to put together, but with PacBio and optical maps, we were able to assemble to the pseudomolecule level these maize genome assemblies. And so we published a couple of years ago 26 de novo assemblies of maize. And then we were able to look at a new type of diversity that exists within the maize genome. So maize is an ancient polyploid, and so we were able to dissect the genome evolution after this polyploidy event. So about anywhere between 5 and 60 million years, it differs based on what paper you read, there was a allopolyploidization event, meaning that 2 different species hybridized and formed what became maize. And so within the maize genome, you see 2 co-resident subgenomes, which we'll call M1 and M2, which are the genomes for this ancient species that hybridized to form maize. Sorghum, a closely related grass, didn't have this polyploidy event. So it only has 1 copy of the typical, what's called Androbotania genome. So if we look across comparisons between the sorghum genome and the maize genome, if you go up in this first column of this plot, you can see for all the 10 chromosomes of sorghum, if we focus on chromosome 1, you'll get 2 copies of a sorghum chromosome within maize in different locations of the 10 chromosomes within maize. So that's the legacy of this polyploidy event. And you can paint across the 10 chromosomes of maize those 2 ancestors, maize 1 and maize 2, across the genome. And so after polyploidy, there's really a couple of different outcomes that could happen with these duplicated genes that exist within the maize genome. One is that there's no change. Both of the copies of a gene are retained. Another is, since there's redundancy, you could get neo-functionalization or diverged function of one of the copies of the gene because the other copy can retain the original function. And then the third outcome is that there could be loss of that second copy because it's redundant, it's not doing anything, it's a waste of transcriptional space, so let's just delete it from the genome. And so we thought that these processes probably played out shortly after the polyploidization event. And that there wouldn't be very many differences if you looked within a population of maize in terms of the types of deletions or neo-functionalization that you could see across individuals. Until we got these sequenced genomes and started to take a look at patterns of these deletions that are called fractionation across individuals. And what we saw is when we looked at tropical maize, which would be all of these CML lines and the KI lines, the NC350 on the bottom right there, they shared a lot of these deletions within the tropical population. And then when you compare temperate lines to other temperate lines, they shared a lot of the deletions. But tropical and temperate lines had separate set of these deletions that were ongoing after this polyploidization event. So that suggests that there's actually population-specific patterns of fractionation and that this process of fractionation could be ongoing even to the present day, even though that polyploidization event happened around 12 million years ago. And so my student Sam Snodgrass took a wider look at this fractionation process. So we assembled genomes of all of the wild relative species within the genus Zia, and then also individuals from the Tripsicum genus, which is a sister genus to Zia. It's about 650,000 generations diverged. And then we compared the extent of shared deletions within these genomes and just the overall extent of shared synteny between these genomes as well. And just looking at that, really large-scale synteny, we already started to see some really interesting trends. So remember, sorghum is that species that hasn't undergone this whole genome duplication. When we compare its genome to the Tripsicum genus, which shares that whole genome duplication with maize, but is about 650,000 generations diverged, we can see that there are 18 chromosomes of Tripsicum, and those correspond almost identically to each of the 10 chromosomes, 2 copies of those from sorghum. And so if you follow these riparian plots of the sorghum chromosomes down to Tripsicum, you can see that there's 1 copy of a sorghum chromosome in Tripsicum on 1 chromosome, another copy on another chromosome. So there hasn't been a lot of fissions and fusions and rearrangements of the ancestral sorghum genome within this Tripsicum dactyloides. But when you go to maize, which is reduced down from those kind of 20 ancestral chromosomes in the polyploid back down to this 10 ancestral chromosomal number, you see this complex patterns of fissions and fusions, really dramatic rearrangements within the Zia genomes, all the different Zia species that's happened just over the last 650,000 years. And so Sam was interested, okay, we see these really big differences between these genera, Tripsicum and Zia. Do we see differences, beyond just these syntenic differences, do we see differences in this genome downsizing and these deletions after polyploidy? And so she looked at the 2 subgenomes, M1 and M2, and then she looked at, different patterns of whether or not, genes were fully retained across both subgenomes. Were there particular deletions that were unique to M2? Were there particular deletions that were unique to M1? And in what instances would we see both copies of these genes retained? And what she saw, first result, that in the M1 genome, this ancestral genome from one of those species that contributed to the hybridization, that there's this preferential retention of genes from that M1 genome. And there's kind of biased fractionation or biased deletion when we look at this M2 genome. So basically, one of the species that hybridized to form maize has gotten its genome beat up and deleted preferentially. So it's this process called biased fractionation. And then she also noticed an interesting result where when she looked at just trypsicum, there was a bias towards more retention of both of those copies from those ancestral genomes within that trypsicum genus. So what that suggests is there's something that separates, just within that last 650,000 years, these two genera, such that there's a quiet genome that's retaining a lot of those duplicates from the ancestral polyploid. And then there's this really active genome within the Zia species, where we've got complex rearrangements, fissions and fusions, and much more deletions of these duplicated copies of genes post-polyploidy. And so we've really had two pulses of deletions following this whole genome duplication. The first occurred right after this whole genome duplication event, and by and large, that was most of the deletions, happening right after that whole genome duplication. But then, after the divergence of these two genera, we have another pulse, basically, of deletions, with trypsicum being a quiet genome that retains a lot of those copies following the whole genome duplication, and Zia having a pulse of deletions after that split. And so what does this all tell us? The main conclusions, in looking at these genomes, these genome assemblies, patterns of synteny and gene loss after polyploidy, we see strong differences between this trypsicum genus and Zia in fractionation outcomes, possibly related to these different chromosomal rearrangement histories. And then we see that most fractionation occurs soon after the whole genome duplication, but then there's another pulse right after the divergence of the trypsicum and Zia genera. And so looking forward, some things that we want to have a look at is, looking at these differences in fractionation across species, across these genera. How has population size of these different species and the genera affected rates of fractionation? Has differential fractionation been important for the unique adaptations of all these species, particularly within Zia? And how strong has selection been for smaller genomes? For example, Mexicana, which is up at high elevation, has smaller genomes. And do we see more extensive deletion of these duplicated copies within Mexicana? And so I'll end there and acknowledge my collaborators on the PanAn project, which generated a lot of the genomes for this. And then also the folks in my group, Margaret Woodhouse, Ren Seathram, and Samantha Snodgrass, that did pretty much all the work that I presented. And I'm happy to take questions. Yeah.
You talked a lot about neo-functionalization and fractionation, but I was wondering if you found any evidence about self-functionalization across the retained duplicates.
We actually really haven't got a chance to look at that yet, and it's a little tricky because there's lots of genomic resources for maize, but in terms of gene function, there's still a lot of domains of unknown function. So it would take a lot of really careful studies to try to get at gene function before we could effectively do that. Yeah, good question though. Yeah.
Hello, everyone. Thank you for coming to attend my talk. My name is Hannah Verdonk. I'm a PhD student at Temple University, and today I'll be discussing my work in investigating selection pressures on synonymous substitutions using a novel codon evolutionary model. So when we talk about synonymous substitutions, what we're talking about is a substitution which changes a genome sequence without changing the encoded protein. And despite the common wisdom that synonymous substitutions are selectively neutral, there's substantial evidence that there is a lot of purifying selection acting on these substitutions for things like translational efficiency, mRNA folding and stability, and CpG site maintenance, in particular for things like methylation. And this substitution can lead to biased codon usage across the genome, and can lead to differential rates of substitution for different pairs of synonymous codons. Up here you'll see a toy example. Isoleucine has 3 synonymous codons, and the arrows between them are ideal rates of substitution. So ATA to ATT is much faster than ATT to ATC. And these different rates could point us in the direction of what selective pressures are acting on these synonymous substitutions, because it takes longer for a substitution to rise to fixation if it is under selection. The problem with that is that existing codon evolutionary models aren't really capable of studying selection on synonymous codons and their rates. And I'll give you a little topical example since the Olympics are happening right now. Imagine you went to an Olympic track event and you wanted to say something about what makes the fastest athletes the fastest. You wanted to know, is it their training regimen? Is it their height? Is it some third factor? But the only measurement you had of these athletes was the average total speed of all the athletes on the track. It might make it a little difficult to say anything about individual groups. So let's give each group its own rate. So traditional codon models have a single rate of synonymous substitutions, DS. This is the average rate of all the athletes on the track. I'm working with multi-class synonymous codon models, which allow you to create different rates for different groups. Prior work in my lab has started using these models in real systems. We wanted to see just what is the effect of selection on synonymous codon usage. And so we looked at the Enterobacteriaceae because we knew that selection on translational efficiency was likely to be a very dominant selective pressure. And so we hypothesized that if selection on translational efficiency was dominant, then it would be strongest in genes with high expression and weaker in genes with low expression. And likewise, codon usage would co-vary most strongly with that selection pressure. So we looked for codons that were evolving more neutrally, codons which did not co-vary strongly with selection pressure and with gene expression. And we found that alanine and valine were the least correlated with this gene expression. We ended up placing alanine and valine into a neutral set and all other synonymous substitutions into a selected set and estimating a synonymous substitution rate for both sets. And what we found was that relative to this neutral set, there was weak purifying selection operating on these selected synonymous substitutions. And that was very interesting, but it got us thinking. How did our choice of neutral set affect the model's estimates of the rates? And were those rates equally valuable and equally valid for all genes? So to start answering that question, I've developed an expanded MSS model. Now, instead of just 2 different groups, we have 67, one for each pair of synonymous codons reachable by a single nucleotide substitution. So here, with our isoleucine example again, each arrow is its own rate. Now, we took this model and we took about 11,000 Drosophila gene alignments, and we fit this model to each alignment and estimated 67 rates for each gene. And this is the result of that. I'm going to walk you through this slide because I know it's a lot. So along the x-axis is each of those 67 different rates. Each column is a heat map of the rate estimates for that rate for all 11,000 genes. The color density is the number of alignments with roughly that rate estimate. You see along the y-axis, that's the relative synonymous substitution rate. The bar across the middle at 1 is relevant because these rates are estimated relative to each other, and they are constrained to have an average of 1. So rates which tend to be faster than 1 tend to be faster than average, and rates that are slower than 1 tend to be slower than average. Each of the little black tick marks is the median rate for that particular estimate. Now, there's a couple big takeaways right off the bat from this slide. And the first is that one model does not really fit all genes. There's a huge diversity of rate estimates per rate and also per gene. Not all synonymous substitutions occur in all genes. You see these little red bubbles along the bottom. The size of the bubble corresponds to the number of genes for which the rate estimate was so low it was effectively 0. We simply did not see that substitution in that gene. And likewise, for some rates, they occur very quickly in some genes. Again, the size of the bubble at the top corresponds to how many alignments had that particular fast rate. Now, going back to the speed of different synonymous substitutions, some of them are occurring faster than average, faster than their fellows, and some of them are occurring much slower than average. And so we thought to ourselves, is that really reflecting different synonymous substitution pressures? So to answer that question for ourselves, we looked at an independent data set of selection on synonymous substitutions, which was a Drosophila melanogaster population genomics sample. We reasoned that selection on synonymous codon usage is mostly purifying, so we expected to see fewer SNPs at codons under selection because variation is less tolerated. So in addition, when there is purifying selection, there is lower SNP density and slower MSS rates. And when there is no purifying selection, there is higher SNP density and faster MSS rates. And we do see that there is a significant correlation between these two, which indicates that, yes, these rates are a good measure of selection in our data set. So from there, we asked ourselves, okay, we can detect selection. Can we detect specific selection pressures that we know are acting on our data set? So we went back to our translational efficiency hypothesis. We reasoned that because translational efficiency is selecting for codons which match the largest pool of tRNA acceptors, then substitutions which went from a very large pool to a very small pool of tRNAs were likely to be disfavored and have slower rates. And likewise, substitutions between codons with roughly equal pools of tRNA were likely to be faster and more neutral. So we calculated the abundance ratios in tRNA pool size between each of the codons in our substitution rates. And again, we correlated them. And again, we saw a significant negative correlation. Now, in this case, we're not surprised that the correlation is only moderate because we don't expect that selection on tRNA abundance is the only selection pressure acting on synonymous codon usage. And going forward, I'd really like to start teasing out some of the other possible effects on synonymous codon usage, like mRNA folding and stability. But to recap, I've explained to you the MSS model, which is capable of detecting this synonymous rate variation across different codon-codon substitutions. These MSS rates correlate with independent measures of selection on synonymous codons. And it's capable of recapturing known selective pressure on tRNA abundance and efficiency. Going forward, as I said, I'd like to start looking at other selective pressures on synonymous codon usage. I'd also like to bring this model to other data sets, other empirical data sets, because we've seen already that these rates and these selection pressures vary depending on which species you assign this to. I'd like to thank my lab, my advisor, Dr. Sergey Pond, as well as my collaborators, and I'm happy to take any questions. So you asked, just to repeat for the camera, you asked if I looked at what was happening to the rates when a gene was under positive diversifying selection. Not yet, primarily because I was just testing to see if the model would work, but that is an important avenue that I would expect, mostly because positive selection isn't super common.
Do you have any intuition on how universal you think your results would be? So when you do apply it to other maybe non-model organisms, do you have a sense of variation, what you expect?
So Applying this model to other systems, non-model organisms, I would expect that the specific rates would look very different. I've already applied this to several other model organisms and seen very different rates between all of them. But I would expect that faster rates were typically indicative of more neutrality.
So my name is Alex Moffett. I'm a postdoc at Northeastern University in Boston in the Center for Theoretical Biological Physics. And I'm going to talk to you today about my work on gene duplications. So we heard something about this a bit earlier, but gene duplications can lead to one of the copies getting a new function, a copy losing its function, both copies staying about the same, or sharing function, and so on and so forth. And oftentimes one copy is conserved while another isn't. And a long-standing question is, why is this? What determines which copy is conserved and which isn't? Which, as physicists would say, what's breaking the symmetry? So one hypothesis that's been tested some, to my knowledge, is that genomic context is what can influence this. So basically, if in this example we have a two-chromosome ancestor, and in the lineage leading to organism A, there's a 3-gene duplication that's inserted from into chromosome 1, copied from chromosome 2, and this happens after a speciation event that's leading to the two organisms we're looking at. And if we look at, if we line up these two genomes and draw a dot whenever two protein coding genes are homologous, and we look for conserved gene order, shown in red lines here, we can see that the largest block of conserved genes, or the largest sort of microsynteny block, is the one where we have the original context, as opposed to this smaller block, which I've labeled as out of context. So this is for the 3 genes highlighted there in organism B. So we can actually use synteny, defined in the microsynteny way, not in the other sort of definition, to figure out whether genes, which copies of which genes are in the original context and which are in a new context. But this is only actually the case if this duplication event happens after speciation between the two species. So that's on the left-hand side shown. It's the same thing I showed you last slide. And on the right, if there's a duplication before speciation, we can no longer really tell from synteny alone which is the original copy, because it looks like we have, we have two candidates and we can't really tell which is which. So this is a problem that I'm going to sort of sweep under the rug for now. But this is something that we can look for largest synteny blocks, and then after the fact sort out when the duplications happened. So we wanted to test this hypothesis of genomic context determining the conserved copies of genes from a duplication on a large data set using this method. So we have 193 mammal genomes, chromosome length assemblies, in a single bird outgroup. And we determined homology between all protein coding genes and determined synteny between all these genomes, and then also did self-comparison. So compared genome with itself and looked for conserved gene order, which will tell us something about where large duplications, multi-gene duplications are happening. So there are a bunch of ways to detect microsynteny. Microsynteny, again, the loosely defined concept of sort of mostly conserved gene order in a region. And there are a lot of dynamic programming algorithms that take a scoring system and give you back conserved gene order. I tried those out and wasn't too happy with the results because I got a lot of weird, like really strange results. So what we've done is gone for a much simpler approach and just looked for perfectly conserved gene order, which we call nanosynteny. Yeah, we don't need a scoring system for that. We can kind of just look and find it. It's very easy to find. And this sort of gives us a low false positive approach. We wanted to also make sure that we weren't getting randomly appearing blocks of conserved gene order. You can imagine if you have a very high false positive rate in homology detection, it can look like, you know, a bunch of genes have conserved gene order, but that's just due to random chance. So what we did was we took genomes and we permuted them randomly. So in this case, I've shown for mouse and human, I randomly permute, get to more and more random dot plots between the two genomes, and look at how the number of synteny blocks decays. And actually, if we look for pairs of genes that have, that are next to each other, we find a sizable number that occur just due to random chance. Blocks with 3 or more genes don't occur due to random chance. And we did this for all, 19,000 pairs of genomes, and we found the same thing. So we're pretty safe if we go with synteny blocks of 3 genes or more. So going forward, okay, here we go. So then we applied our method, right? We just looked for the size of the synteny blocks that our genes were in. So in this case, we take organism B as our reference species, and we go through gene by gene and look at the genes that are homologous to that in organism A in this example. And then we measure Levenshtein distance to tell us how similar the two sequences are. And then we can label when we get to the largest synteny block, if this gene is homologous to 2 genes that are in regions of conserved gene order. And keep going, and what we expect basically is that the distances will be lowest for genes that are in larger synteny blocks, meaning that are in their original context. And okay. So here's an example again of what we actually, the actual analysis. This is comparing mouse and human, and we find that actually 95% of these human genes, there's fewer genes than you'd expect because we're only looking at genes that are in some region of conserved order, at least. And 95% of these human genes, this is the case, where the longer synteny block gives us the more similar genes. And then, so here's the result for a bunch of other examples of pairs of species. And we see that, high 80s to low 90s% of genes, this is true. If we then restrict ourselves to only looking at genes that are in more than one block of conserved genes, meaning where there really has been a large duplication, multiple gene duplication, this number now goes down to like 47% of genes in some cases to 94% in other cases. So we think this is probably because of that problem that I mentioned at the beginning, where we really need to differentiate between cases where the duplication happened before and after speciation. So just to summarize, we found that homologous gene pairs in synteny are in fact much more similar than those not in synteny. And in general, homologous genes that are in larger nanosynteny blocks are more similar than those in smaller blocks. And yeah, we're working to differentiate between the two different cases I mentioned. And also do a bunch of other analysis on this data, including some DNDS analysis. So maybe I should look at that, look at your work there to get a better model. And I want to thank Michele Di Piero and Andrea Falcone, who I worked on this project with, and also the DNA Zoo team, who's cranking out genomes left and right. So yeah, and thanks for listening. Any questions? Yeah.
Yeah, it'll definitely be interesting to look at DNDS with those duplicates. And I'm curious, since it's human mouse, I'm assuming there must be all sorts of expression data sets. And it'd be interesting to look at levels of expression in the duplicates and levels of tissue specificity in the duplicates.
Yeah, exactly. We're also wanting to look at epigenetics, and so there's a lot we can do. At least with those two, obviously, the most of our data we don't have that for, but mouse and human, yeah.
Do you have any sort of suspicions about what the syntactic context that's mattering here is? Is it sort of the regulatory environment or the confession involving the chromatin?
Yeah, I glossed over that. I forgot to mention. Yeah. So yeah, I think it's probably cis-regulatory elements that are most likely. Yeah, that's probably what I think. But we also it could also be the 3D genome context, and we actually have high-C data for all these species. So yeah, we could look at that.
Cool stuff.
Huh?
Very cool stuff.
Oh, thank you. Thanks. Any other questions? Oh, yeah.
Were the synthesis regions evenly distributed around the genome? Do you know?
That's actually, I haven't actually looked at that, surprisingly. That's a really good question. Probably, I mean, the sort of, I think the accepted thing is that there are hotspots for breakpoints in genomes. So I would guess not. It would not be evenly distributed, but I need to look. Yeah.
This is the last talk of this session, so I'm pretty sure you can all stay for as long as it takes to get through this. So would you believe that female orb web weaving spiders that make this iconic wheel-shaped web produce 7 distinct silk types, functionally distinct silk types? And they can do that because inside of their abdomens, they have 7 morphologically distinct silk glands from which they produce each silk type. So just to show you a few, 3 of the 7. First, in yellow, we have the major ampulate gland that produces the silk that the spider uses to construct the frame of the orb web. Then here in blue, we have the flagelliform gland. And the spider makes silk from that gland to produce the elastic capture spiral of the orb web. Here in green, we have what's known as the aggregate gland, which produces a wet glue silk that coats the elastic capture spiral to make it sticky so that insects adhere when they hit the web. And so that's 3 of the 7. And these are stress strain curves for some of those silks to show how they are different in their mechanical properties. So how much strength, extensibility, and toughness each one exhibits. And they need to have these different properties so they can perform their various functions. So we know that the major ingredient of these different silk types are members of a class of proteins or a protein family encoded by a single gene family known as spidroins. And spidroins have a common architecture. So the proteins, here's a schematic of one shown here, are usually very long and very repetitive, highly repetitive. So we have these non-repetitive short terminal domains that cap it, but most of the length of the protein is essentially composed of a particular amino acid sequence that's repeated over and over and over again throughout most of the length of the protein. So these repeats can often be decomposed into amino acid motifs that have been characterized biophysically. So it's shown that different ones conform to different secondary structures that give the proteins that compose a particular silk fiber type their mechanical properties. So some have a lot of beta turns, which would create these nanosprings that give a particular silk more extensibility. And then some have a lot of beta sheets, which form these nanocrystals that give strength to a particular silk type. So all of the spidroans have this common architecture of being long and highly repetitive. But when we look at the different spidroans that make up the different silk types produced by this one spider, they vary tremendously in their repeat sequences. So here we have the 7 silk types made by a female orb web weaver. And again, here's the common architecture. And next to each silk type, you see a box here with an amino acid sequence, which represents the repeat type associated with that protein that makes up that silk type. And I just want you to observe how the repeats vary tremendously in terms of their length and amino acid composition across the different silk types. And so it's thought that this repeat variation is what's basically explaining the great mechanical divergence that's exhibited by the different silks that this spider makes. And it's been driven by a series of gene duplications that happen throughout the history, the long history of spiders, long evolutionary history. Now, what I showed you is really well understood for orb web weaving spiders, and particularly those in this clade known as the Araneoid orb web weavers. Now, it's a big clade and it's got a lot of families, and in fact, we really understand the system very well from just a small number of species in 3 families depicted here by these red arrows. And it turns out all spiders make silk, and they do so by drawing on different proteins from the same family that I just told you about. And this lovely cartoon over here depicts some of the diverse web forms made by all the different kinds of spiders we have out there. And a lot of this diversity is unexplored to date. So for today, I'm going to tell you a little story about one branch of the tree represented here by a family of spiders known as the Dinopidae. So the Dinopidae are known as netcasters. Okay, so here's a little movie to show you the action of the Dinopidae. So they build a web, this fuzzy web that they hold between their front legs, hanging upside down. They are waiting for some insect that then they'll stretch out their legs widely, stretching out the web, and then throwing it over their insect prey, okay? And I want to point out that this web here, here's the capture threads of the web magnified. And further magnified over here, in fact, this fuzzy silk is, it's a composite that's woven together from 4 different silk types that are produced from 4 different glands in this spider. Okay, so that's amazing. Now, all right, so this web here is actually thought to be potentially homologous to an orb web. And what I'm showing you here, in fact, this spider makes 8 different kinds of silks if it's a female spider. And I have the 8 different types listed here and an icon to represent essentially what it's doing functionally. So here's the web, and here's a magnification of that fuzzy capture silk. Okay, so yeah, it's sort of, I should mention, it's different from that web I showed you at the beginning that had the wet glue. So this is a dry adhesive, much like Velcro. All right, so here we have this fuzzy silk, and 5 through 8 here are the 4 different silk types that are woven together to make this capture silk. And 6 and 7 here are 2 silk types that are thought to be ancestral silk types. So essentially, these are silk types that evolved in earlier spiders that were retained in the dinopids, and they were lost in those araneoids that I mentioned. And then here, number 8 is what's known as the pseudoflagelliform silk, which is this line right here. And you can think of it as like the platform on which this fuzzy silk is built, this fuzzy capture silk. So it's thought that this pseudoflagelliform silk is homologous to the flagelliform silk of the aranioid orb weavers that I mentioned at the beginning that is used to make that elastic capture spiral of the orb web. So, there have been many recent studies that have attempted to resolve the relationships of this interesting family Dinopidae with other major spider lineages, such as the aranioids and others shown here, various phylogenomic studies. But because these are very deep splits, essentially, it's been difficult to